Okay, so on this next slide, this is talking about the neuromuscular junction. So neuromuscular junction means where the muscle and the fiber, where the muscle fiber and the nerve cell meet. So it's a type of synapse, meaning a connection. It's also called a myoneural junction. But see here, I'm going to underline site where the axon. Axon means the end part of the nerve cell. And so we learned how the neuron means a nerve cell. And if it says a motor neuron, I'm going to write this at the bottom. Motor neuron just means the nerve cell that goes to activate the skeletal muscle. It activates a muscle. But yeah, so again, this is just the site where the neuron and the muscle fiber interact or meet. And so on this picture, this yellow part, this is the end part of the nerve cell. It's called the axon. So the axon has these extensions or these branches at the end. The very ends of these parts are called the axon terminals or the synaptic knobs. And so then in this picture, this is showing a close-up view of one of these axon terminals or the synaptic knobs. And so then here it's interacting with the muscle. And the muscle is the bottom part. Skeletal muscle fibers contract only when stimulated by a motor neuron. Okay, so then these are basically the steps for the contraction that the video laid out for us. Now, we have to know that acetylcholine is the chemical that's released by the nerve cell. So the chemical released by the nerve cell is called a neurotransmitter. This is just a general name. There are many neurotransmitters, but so acetylcholine is one of these neurotransmitters. So this is only released for the skeletal muscle contraction, but also it can be used for by the parasympathetic nervous system. So here, nerve impulses that come down the nerve cell. And when we get to the nervous system, we're going to learn about these nerve signals. So first, these nerve signals causes the release of acetylcholine from the nerve cell. Then the acetylcholine goes to the muscle. So on the muscle, there are these small proteins called the acetylcholine receptors. This is where the acetylcholine attaches to the muscle. Okay, so once acetylcholine attaches to the muscle, then the acetylcholine causes an electrical signal in the muscle. Electrical signal is also known as an action potential. Then the signal causes the release of calcium from the sarcoplasmic reticulum. Okay, so now that we have our calcium, then we can go to the next step. Okay, so again, we're back to this. If there is calcium around, calcium is able to bind to troponin. So basically, I kind of think of it as like calcium pushing the troponin button. Okay, so once you push the button, then the tropomyosin is going to move. And so here, the tropomyosin, it used to cover those binding sites on actin, but now it moves away. Well, now those binding sites on actin are open, and the myosin can bind to actin. Okay, so again, here this is just showing on the troponin. Troponin is where calcium attaches. And once calcium attaches to troponin, tropomyosin moves away. And now those binding sites on actin are open. So you can see those little spots are open now. And myosin, which they're not showing on this picture, they're not showing the thick filaments, but now myosin can attach. Okay, and the slide is then showing the sliding filaments contraction, and so basically showing the cross bridge. Cross bridge just means when the myosin and actin are attached. Myosins attached to actins. So of course there are many of these myosins, many of these little actins, so they're all attaching. Okay, so this top part, that's a thin filament. So here, this is just showing after calcium has come and tropomyosin has moved away. So here, those binding sites on actin are open. And myosin can now attach to actin. So again, myosin cross bridge, meaning myosin attaches to actin. Next thing, as soon as myosin attaches to actin, myosin ends up bending its head, and when it does this, it ends up pulling the thin filaments towards the direction of the bending. Okay, when this happens, ADP and a phosphate is released. So when it's doing the pulling, when the myosin pulls, this is called a working stroke, also known as a power stroke. Okay, now it pulls, but it's still attached here. In order for myosin to detach from actin, in order for it to let go, an ATP molecule has to come and bind to myosin. So when ATP comes in, the cross bridge detaches. So here ATP attaches to the myosin, and myosin now lets go of actin. Okay, now, but here, after it detaches, myosin is in this low energy level, okay, because it used up the energy to do the pulling, so now it needs to get back in its upright position. Okay, so then in order for it to get to its upright position, ATP needs to be split into ADP and the phosphates. So once it does this, they call this the cocking of the myosin head, but it just means when the myosin's head is upright. So now it's ready for another contraction. As long as the actins are open, or those binding sites are on the actins are open, myosin can do this again. And in order for the myosin or the actin sites to be open, calcium still needs to be here. So as long as calcium is available, myosin can still go and attach to a new actin and pull that. Let go, attach to a new one, and then pull that and let go. Okay, so this continues on and on. Okay, so this is what a contraction is. But then how does the muscle relax? Before we talk about the relaxation, this is just another slide just showing the same stuff as the last picture. And so it just goes through how the myosin head attaches to the actin. This is called the cross bridge. Myosin pulls the actin, or in other words, the thin filament, towards the center of the sarcomere. When this happens, an ADP and phosphate are removed from myosin. New ATP needs to attach to myosin in order for myosin to let go of actin. And then, to re-energize myosin to get it back into its upright position, ATP needs to be hydrolyzed or split. Yeah, so this is called the sliding model of contraction. So during contraction, a sarcomere is getting shorter because the thin filaments are being pulled towards the middle by the thick filaments or the myosins. So what it's saying here in this top picture, it says this is showing the relaxed version. So the thin filaments are not all the way in the center. But then the next one is showing contraction and then contracting even further. So again, we're just seeing those thin filaments moving towards the center. And I'm going to write down here, thick filaments remain in place. So they stay in place, but it is the thin filaments that move or are pulled towards the middle. Towards the middle inside its sarcomere. Okay, so then during contraction, those regions are changing in their size. It says here the H zones and the I bands are getting narrower. So if you remember, the I band meant the part where you only see the thin filaments. So here you can see how this is shorter, smaller, this distance, than when it was relaxed. And then also the H zone, where you only saw the thick filaments, that's also getting smaller. And the Z lines are, of course, moving closer together, since the thin filaments are moving towards the center and the thin filaments are attached to the Z lines. Now, the thin and the thick filaments are not changing their length. It's just the location for the thin filaments is changing. They're just moving towards the center. And you can notice here how the size of these lines, the thin and thick, are not changing at all.